Hello, everyone. Thank you, Katie, for that great introduction, and thank you all for joining us today. And welcome to the NOAA Omics Seminar Series. I'm your host, Nicole Miller, the NOAA Omics Coordinator, and today's presentation is brought to you by NOAA Omics and NOAA Library. Thank you very much, Katie. Let's go ahead to the next one. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. The seminars from the series will be made available on the NOAA Omics and NOAA Library YouTube, as well as the NOAA Omics landing page. So with that, today our presentation is titled Detection and Relative Quantification of a Dungeness crab from Environmental DNA or eDNA samples using quantitative PCR by Dwan Jackson. Dwan is a NOAA CCME graduate scholar at Jackson State University, and through the EPP MSI program she is a part of, she completed a 10-week long internship program at the NOAA Pacific Marine and Environmental Laboratory within the Ocean Molecular Ecology Group and completed this project under her mentors Dr. Zach Gold, Dr. Sean McAllister, and Han Wyrick. Welcome, Dwan, and thank you for presenting today. I will now pass it off to you.
Thank you so much for the presentation. I'm going to turn off my camera just so everyone can see all of my slides. And thank you so much for letting me have this seminar. And again, I'm going to be talking about my project, the detection and relative quantification of Dungeness crabs or Metacarcinus magister from environmental DNA using quantitative PCR. And this was again done during my NERTO here at NOAA's PMEL with the Ocean Molecular Ecology Group. And I'm going to want to start and explain a little bit about the a little bit about the background about the species, about why Dungeness crabs is our particular species of interest. So for our economic significance, Dungeness crab, also known as Metacarcinus magister, and I might possibly refer to them as Dungies throughout this talk, but they are very important economically due to the fact that they bring in a little over $222 million annually and is considered one of the most valuable edible crab commercial fisheries found here on the West Coast. And this high value makes increasing concerns of the economic uncertainty this species is facing due to the fact that there are increasing threats due to a variety of different environmental factors like ocean warming, ocean acidification, hypoxic events, and more, which affect the fisheries and the harvests that are returned from it. Along with that economic significance, we also have their ecological significance. So Dungeness crabs are found here on the west coast of North America, starting from Alaska all the way down to Mexico, with the fisheries kind of stopping about mid-California. The adults of these species are typically found on the coastal areas and in small estuaries, and they are considered opportunistic carnivores and benthic scavengers, with diets being primarily composed of fish, bivalves, and brown shrimp. And with that diet, they play a significant role in regulating the population densities of these prey species and also aid in nutrient cycling with that benthic scavenging. Along with their ecological history significance, I want to talk a little bit about their life history because there's a specific life stage that we'll be focusing on later on in this talk. So for their life history, once they are hatched from the eggs, they start off as zoea, and they're planktonic and they drift in the water column and exhibit a positive phototaxis and rheotaxis. That positive phototaxis meaning they're attracting towards the light, and that rheotaxis they're attracted towards water currents. They then become megalopy, which is where we begin to see them move close towards that nearshore environment and estuary environments so that they can begin to settle, and that positive phototaxis and rheotaxis becomes much stronger. And because they're going towards that settlement, they begin to also exhibit thigmokinesis, which is when they begin to cling to objects while they are swimming. Also, this life stage is where we begin to see recruitment into the fishable population. However, we do not fully, at this point in time, understand how this recruitment affects the adult populations. Once they transition, once they have begin to settle, they begin to molt into our instars, and that's where they begin to settle into the benthos. And those instars are also sometimes called juveniles as well. And when they settle there, they are primarily prioritizing areas that provide protection. So your shell habitats, your rocky gravel habitats with macroalgae and eelgrass. And they'll spend about 1 to 2 years here before they migrate into the deeper waters as adults. And of course, the adults, they settle in the coastal areas, small estuaries, and inland waters, and the adult age is determined by its size. With that life history in mind, I also want to talk a little bit about the cultural significance they have to the West Coast and how that this will tie into understanding that that life stage specifically with the megalope. So some cultural ties the species has to the West Coast is important to a variety of different tribes, especially here in Washington. So for some examples are the Tulalip Tribe Alliance, the Port Gamble Slalom tribes, and the Swinomish tribe, which is who I worked with for this project. And a lot of these tribes have a dedicated catch period before the season opens due to their extensive history with it, with the species, and also are exempt from a lot of the commercial fisheries, commercial fishery harvest requirements. Also on the West Coast, a little bit of fun things. So in the top left here, I have a picture from the Pike Place Market here in Seattle, and it's kind of small and a little cut off, but they're selling Dungeness crab legs here along with a bunch of other crabs and seafood here. And then also even in Washington, at the bottom, there is a Dungeness crab festival where essentially everyone gets together and eats a lot of Dungeness crab. And then in San Francisco, it is considered a Thanksgiving tradition to eat Dungeness crab as opposed to turkey. So along with that cultural tie, so it's deeply ingrained within the culture of the West Coast. So much so that a research group called the Pacific Northwest Crab Research Group was developed, and they bring together tribal groups, local communities, academic institutions, and government agencies, and they come together for the purpose of producing research to inform effective fisheries management. And this is done with Dungeness crab being held as its main species of interest. So all this all encompasses just how important Dungeness crab is, not only economically and ecologically, but also culturally as well. And how I talked earlier about that larval recruitment, I also want to go into a bit about that, how the species is managed and why larval recruitment is so important to understand. So it's currently a state-managed fishery in the terms of done being on the 3S principle of size, sex, and season. So the Dungeness crabs can only be caught at a certain size because it assumes that they have at least reproduced at least once. Males can only be kept as the females need to be released to ensure the population's replenishment ability, and it must be caught during the Dungeness crab season. And the way we are recording all these catches is through self-report catch data in the terms of mandatory monthly logbooks or catch record cards. And currently, this overall management has been deemed to be moderately effective. So all this in turn has been, while this fishery has been deemed to be moderately effective, it has also been deemed to be reactionary in the fact that it is unable to adapt to environmental issues leading to problems like economic uncertainty and possible unsustainability due to these economic factors as well. The way we are currently estimating the abundance of the fishery is through depletion estimators. And this estimates that preseason abundance based on that fishery-dependent data, which only records for the caught legal size males. And this is also deemed to possibly aid in that reactionary approach because it tends to miss a key component, which is the larval stage, which is why it's so important to understand it, because that's how we know what we're getting into the fisheries. So when we're only using the outputs of the fishery, to estimate our preseason abundance, we could possibly be over or even underestimating our abundance. And so not having that larval stage is very critical. And also, without having that input of the larval stage and their recruitment, we are not able to do a formal population assessment, which helps indicate the stock health and sustainability of the fisheries overall, which is becoming increasingly concerning, also due to the fact that we cannot account for recruitment variability because it can be affected by a variety of different things like environmental factors or essentially season disruptors that can cause the season to open later, and which in turn will affect the recruitment process. And I mentioned the megalopy earlier and why and now we can see why understanding this larval stage is very important, and also especially the recruitment as well. So when we're looking at the megalopy, here's a really zoomed in picture of the megalopy with practically the phone right up to the jar. The megalopy are about the size of your pinky nail, and the way they're distinguished in the field based on from other megalopy under other cancrids megalopy is due to the coloring of their body and their larger size difference. However, there are reports of a smaller size of the Dungeness crabs due to hypoxic conditions or their phenotypic origins. And the reason why understanding this megalopy in particular is important as opposed to zoea is because of the critical period hypothesis. So the critical period hypothesis basically states that the larvae need to find suitable environmental conditions and food in order to survive, or else they will die off. And it seems that for Dungeness crabs, majority of that mortality is happening in the eggs to zoea. The egg to megalopy stage. So that zoea stage is where they're mostly dying off at. So the megalopy is what is being recruited into the fishable population. And also, and with that being the recruitment indicator, it is important to look at them as well because they are highly sensitive to a variety of different factors, especially as of recent. So I stated the ocean acidification and the pH and hypoxia, and this fit graphic in particular looks at the ocean acidification side. So starting from left to right, from the left, we're seeing that there's dissolution of the Dungeness crab larvae causing structural deformities, so their carapace is getting a lot thinner. And we can see a lot of those ocean acidification hotspots in the middle are where the Dungeness crab typically inhabit and where they're going to be in that megalopy stage trying to settle. And we're seeing dissolution causing damage or missing mechanoreceptors, which allow them to change, to sense changes in the environment. So this makes it more likely for them to die off. So it's very important to look at this stage because of these increasing changes in them. And also, we are able to use these to help predict our future stocks. So if we don't know what's affecting them and how that population size is changing, how can we look at the sustainable harvest of this species? And it's becoming increasingly important to track them. So now we are trying to generate some of that larval monitoring data, but we are also coming into a bit of challenges. So this is being spearheaded by the Pacific Northwest Crab Research Group, and they're focused on generating this larval tracking data for population assessment models. And here we can see this is a picture of a light trap, which was, and this is built off a long-term larval crab monitoring study in Oregon by Dr. Alan Shanks with these light traps, and they're very inexpensive to make. And it was found that in that study, they were able to estimate what the population abundance is 4 years from the megalopy collection. And these light traps, essentially, they take advantage of the Dungeness crab's positive phototaxis, so that attraction towards light. In the terms of, so the top, with the bucket top, it has that foam to keep it above the water column, and it hosts a battery pack that powers that light down the middle with the funnels attached on the inside. And with those funnels, the Dungeness crab are able to swim in. And when that light trap is pulled out of the water, when looking at that cotton piece with the PVC, there are some sieves there that keep the Dungeness crab in and let the water out. However, we're finding that with this larval monitoring method, what comes out of these light traps is not just Dungeness crab. And here is a picture showing kind of what comes out of this, what this comes out of this light trap. And we can see there's a variety of different animals along with our Dungeness crab megalopy. Also, some other challenges with this is the fact that this method is labor intensive, small scale, and time consuming. So and this is due to the fact that small scale and the fact that majority of the sites are only in Washington and with some in British Columbia, and actually getting these larval counts, so have to sit there and count all the Dungeness crab. It must be done in the field because once you preserve these animals, they're no longer that see-through, and you can't see that brown coloring and distinguish them from the other megalopy as well as easily. as easily. And also labor intensive in the fact that it really depends on the megalopi that are caught. So sometimes you could be maybe in the field counting at each trap for about 30 minutes. But if you're getting really, really large numbers, you can be there for a really long time. For example, from our collection site, this one was a sample site that just had a much higher than usual count. And you can see you can practically scoop up the Dungeness from this bucket, and even with subsampling to count these numbers, it still took almost about 3 hours, give or take, and it can take a really long time. And so we want to see, can we use molecular tools to help alleviate some of these challenges with these light traps and possibly help increase the scalability of the sites and also data turnaround. So our solution is to test and validate a novel targeted qPCR assay for magister and apply it to assess abundance at the critical larval stage and the use of eDNA sampling from light trap effluent samples and bulk plankton samples. So before I get into more of my methodology, I want to first give a little overview about why we specifically chose this approach. So specifically with this qPCR, there are two different types of qPCR. You have your cyber green, which basically you have your dyes just floating in, and once your primer is attached, the dye gets trapped in and then it fluoresces. However, your amplification is only as specific as your primers. So we decided to go for the TACMAN approach, which is where we have a primer and a probe, and you're getting more specificity because your primers and probe must match in order for it to attach. And then once it gets extended, that probe kicks off and fluoresces. And that allows that specificity and our quantification. And the reason why we also decided to use eDNA in specific is because we can detect eDNA in an area non-invasively and non-destructively, which is a great advantage. So because eDNA is simply just short DNA fragments from skin cells, bodily fluids, feces, or urine in the ecosystem, and can come from a variety of different types. So it could be collected from water, soil, or air. It allows us to be able to just pull that water out and see if we can get Dungeness crab DNA. And while this methodology is considered almost in its infancy, we're seeing it become more and more established. And here I'm showing just some similar use cases using qPCR for detecting specific species in eDNA. For example, Spear 2020 used targeted qPCR for walleye in eDNA samples for developing a population abundance tool model for bolstering current management. And we're beginning to see more of this eDNA and qPCR approach being adopted as a monitoring tool and has already shown changes in conservation management and policy. So for this study, our main goals were to test and validate the assay using best practices defined by Thailender et al. 21, also evaluate the effectiveness for quantifying larvae from discarded ethanol, and also apply this assay to real-world samples to further evaluate and demonstrate its utility. This next slide is just a little bit of a table of contents for my methodology. And so first we started off with the assay creation. This was done by the Ocean Molecular Ecology Group, and where I mainly come in is with the testing and validation of this assay, the plankton megalopy jars, the eDNA water effluent collection, and the assay application on eDNA samples. Starting off with the assay creation, a bit of a high-level overview of it. So the assay creation started off with DNA sequences of our Dungeness crab and some closely related cancrids, along with a few outgroup cancrids because they also co-occur. And we generated mitochondrial genome sequences and annotated them. We then used UniqueSeq to develop our candidate assays. And then where I mainly worked on was the testing and validation of that assay. A little bit about, and you're probably thinking like, well, why do we need to develop all these mitochondrial genomes when there's already plenty of CO1 sequences for these different cancrids out there? So while CO1 is the most commonly sequenced, looking here at all these gene sequences, at the top, those are two outgroup crabs, and all the rest from those two are all cancridate crabs, and we can see that their gene orders are fairly similar across the entire cancridate family. So just having that CO1 data, if we cannot find a unique region within it that is conserved to Dungeness crab, we'd have to look elsewhere. So just to save ourselves the time, we generated these mitogenomes so that we could look at some of these other regions to find a specific region that is conserved for Dungeness crab and least likely to get that cross amplification between the other species. So with those mitochondrial genomes, we used UniqueSeq to find a region that targets Dungeness crab and does not target the other 8 closely related cancrids with that one outgroup cancrid. And our longest unique region was found to be the NAD2 region, which is not commonly sequenced, especially for cancrids. And from there, we designed our Tachman assay following Klimas et al. 2020 and Andruskevich Allen 2021 checklist. And from here, we can see when we align our primers and probes that we designed to Dungeness crab, which is at the top, and to the bottom, which is all the other cancrid species, we're seeing we have that highly conserved region for the Dungeness crab, having more than a 2 base pair difference in our reverse, more than 9 in our forward primers, and more than 4 in our probe. And so while for this assay, we wanted to specifically show on our approach of can we get this assay to not only get our absence and presence, but we also wanted to see can we get quantification as well. So using Thallinger et al. 2021, we used their guidelines that they established to test and validate this assay. And these guidelines were generated by analyzing a variety of published qPCR and environmental DNA assays, and create a centralized guideline on how to interpret these assays and validate them. So level 1, the assay is designed and tested on target tissue. Level 2, you have optimized it by testing on the closely related non-target species. For level 3, you're getting testing on eDNA samples, and you have those positive detections with the processing steps recorded in detail. Level 4, you're getting that limited detection along with extensive field testing and in vitro testing on the co-occurring non-target species. And level 5, having the detection and probabilities estimated by statistical modeling. And in order to fulfill each level, you have to fulfill the level before. And our goal with our validation is to hopefully get a level 3 assay, possibly higher. So starting off with our optimization of our candidate assay along with our loading. So we're starting off with the sensitivity, and we're using that NAD2 assay with our synthetic DNA of our Dungeness crab or our gBlocks. And we started with serial dilutions starting from 10,000 microliters, copies per microliter, all the way down to 0.4 copies per microliter. And this was done with 16 replicates for that 10,000 copies per microliter, and then from 1,000 copies per microliter all the way down to 0.4, we did 23 replicates. And here is just showing that amplification plot of that assay. And from here, we were able to get our standard curve and optimize amplification tightness across all of our replicates. And these standards will also be run on every qPCR with a minimum of at least 3 replicates to have referred to with our samples, and also make sure our assay is running properly. With all this data generated, we are able to use this to determine our limit of detection and limit of quantification. And for this, we followed Klimas et al. 2020. And for our limit of detection, it was defined as the lowest concentration at which 95% of the technical replicates had positive amplification. And here we can see that we have 95% about between 2.5 and 1 copies per microliter, showing that we have can detect really low concentrations. And for our limit of quantification, it was defined also following Klimas et al. 2020, and it was determined at which the lowest concentration which the relative standard deviation of back calculated concentrations was less than 35%, and that was looked to be about 100 copies per microliter. Now that we have those data, we also wanted to see what our limited detection, limit of quantification was if we ran through the ELO quant calculations. And these particular calculation uses a statistical model to determine the LOD and LOQ in low copy number eDNA samples. So using that same data, we're able to see that our limited detection, looking specifically at the three number of replicates being three because we ran all of our samples in triplicates, we are seeing that our limited detection is a little less than 0.5 copies per reaction. And for our limited quantification, we're seeing at about 1 copy per reaction, which is really, really sensitive. Now that we have our sensitivity kind of nailed down, then we look to begin testing our specificity. And this was done using tissue extraction of Dungeness crabs and two closely related cancrids, and then our outgroup cancrid, which co-occurs in the same area due to its invasiveness. And we applied the assay there, and we were able to see we got no false positives or cross amplifications, meaning it was very specific for Dungeness crab. Bringing it back to Thallinger et al. 21, we're seeing that our assay has completed level 1 by testing on target tissue and also testing on those closely related non-target tissues. For the rest of our methodology, we're hoping to fulfill level 3 and beyond. Now that we have our assay pretty much tested and validated, we then want to begin starting testing on some real-world well, not necessarily real-world samples, but our next step of the megalopy jars. For our plankton megalopy jars, we're looking at 3 different aspects of it. So our first aspect is mainly want to see, can we get a correlation between megalopy counts, so the number of megalopy in a jar, to the DNA concentration that is released from them. This was done by setting up a series of jars, one jar having 1 crab, one having 5, one having 10, all the way up to about 500. And we put 500 milliliters of ethanol, and that ethanol will then be extracted to see if we can get a correlation between the 2 crab counts. We also wanted to test another aspect, since we were already creating these jars and creating these plankton megalopy jars. We wanted to see, is there a difference between the 2 ethanol exchanges that occurs in plankton extractions? So you're probably wondering, like, why are we looking at these ethanol exchanges? We throw one away. Why does it matter? So typically with a plankton sample, you have ethanol that is removed after 24 hours, and this is due to the fact that water tends to leach from the samples or the tissue, causing that ethanol concentration to change. And so by removing that ethanol and replacing it, you're more likely to have that original ethanol concentration, having better preservation of the sample for the long term. However, because we know that water is leaching, we wanted to see, well, is also eDNA or DNA coming from these crabs as well? So we want to see, can we use this typical waste item for eDNA work, since it's discarded, and also create a new data stream. So we had that ethanol that was typically removed after 24 hours. Instead of tossing it, we stored it, and then we removed the ethanol again about a few days later to roughly replicate what would be a typical ethanol eDNA sample, and those were all filtered. And again, we want to see if we not only get a correlation between crab counts and concentration, but how does it compare from that 24 hours to that typical a few days later, what would be your normal plankton ethanol sample. For our third aspect, we added in some other plankton samples. These were collected during the West Coast Ocean Acidification Cruise in 2016, and these plankton samples have been sitting for about 10 years. And we want to see, well, can we also quantify these crabs as well, but can we also use these older samples to get this similar correlation and quantify the number of crabs in here as well? So from here, for our methodology, for the megalopy jars, for the prepared ones, we remove 400 milliliters of ethanol. But for the WCOA or West Coast Ocean Acidification 2016 samples, we removed all the ethanol. These were all filtered following the OME disc filter protocol to get their eDNA. And then they were applied to the assay to get our qPCR concentration. We also had manual counts of the megalopy so that we can compare and see what the correlation is between the 2. After we applied that, so here I'm showing the results of that DNA concentration compared to those megalopy counts. So on my y-axis, the DNA copies per liter, and for the megalopy counts, I have them at the bottom, and both of these axes are transformed on a log 10 scale, with the left side having that 24-hour ethanol and the right-hand side having that 96-hour ethanol. And we're seeing that we're getting a much higher correlation in that 24-hour ethanol compared to that much later typical plankton ethanol. And we're even seeing that we're detecting much lower number of crab counts, having detections at that 10 crabs per jar as opposed to starting at about 50 in that second ethanol removal. So, and even more so, we're seeing a much greater DNA yield, getting about a 10-time increase in DNA yield, and possibly showing a viable use case of this typical waste product in eDNA studies, and also possibly change the way we collect data overall with that much higher increase in DNA yield. For our West Coast ocean acidification plankton jars, however, we do see we're getting a fairly close R squared to that typical plankton sample, but it's not nearly as tight compared to that 24-hour ethanol. For these samples, however, we did also have to exclude 2 jars, and because they just simply failed. And to go in a little bit about why these failed, you can see here to the right, those are the samples that worked, and to the left, those are the ones that didn't work. And just from looking at it dead on, we can see that there was not proper preservation of these samples, and looks like the different things decompose within it, and definitely did not have preservation of that DNA. And from here, we can see even when we filtered it, it left just a sludge of decomposed animals on the filter, which we lovingly call the bean burrito. So we're seeing that we do get a correlation between megalopy and DNA concentration release. So that looks really good for us for when we begin to start applying this to our eDNA water effluent samples. And so to show more of this assay's application, and especially into improving this larval data, we want to see, can we collect the water from these light traps to get an estimate of the larvae, saving time and money, and also possibly help increase the survey size and data generation. So with the help of Sarah Grossman, she is a Swinomish tribe environmental specialist. We selected 3 light trap sites in Anacortes, Washington, and we went to these sites with Sarah as she collected on June and July. For these different sites, for the light traps itself, as she pulled it out of the water, we stuck a sterile bucket underneath, and we collected 1 liter of water in triplicate, and same for the downshore, where from the light trap, and also at the dock where the light trap was secured to, we collected 1 liter in triplicate of the water samples. We then filtered this through a Sterivex filter and extracted the DNA from those Sterivex filters. And just to overall encompass the total sample size, we have those 2 different sampling dates, the 3 different sites with the 3 different stations at each site for a total of 18 samples, but because we collected in triplicates, that becomes 54 total samples. Now, there are possibly some slight differences between this data due to collection is something I'm currently looking into, but for June, we did use a peristaltic pump, as in July used gravity bags. This is due to the fact that we did not realize how long we were going to be in the field for June, and so we switched to gravity bags to essentially speed up the collection process. So just looking at the light trap and its detections overall, for this graph, for the y-axis, I have my different sites, so the Anacortes, Rosario, and the Cornet Bay, and the stations at the bottom of the dock, downshore, and light trap, along with the megalopy counts to kind of put into context these detections. And we're seeing that for our June sampling, we have detections at that Cornet Bay, downshore, and the light traps, and also that Rosario light trap as well, but we're also seeing high variability between the replicates. And with the July sampling, we're only seeing detections at that Cornet Bay light trap. And we can already see here that we're getting detections really, really well for those really high numbers at that like 4,900. But when we get to the 50, like the 50s on down, we're not seeing any detections whatsoever. Then when we compare this concentration that we got from the light traps specifically to our megalopy count, we're seeing our R squared is not very high, but we're at least getting a positive correlation between the DNA concentration to megalopy. So it looks like for those lower concentrations, this assay may not be very applicable. Taking all this into account, we can see our assay has fulfilled the next two levels. So we tested it on eDNA samples and had those positive detections, and we had our sample processing steps reported in detail. And then we had that limited detection established with extensive field testing and in vitro testing on our co-occurring non-target species, because they do occur within these light traps as well. So some key takeaways about this study. We're seeing that we are getting a great relationship between the megalopy and DNA concentration with the jars, and suggest that we can use qPCR to provide a quantitative estimate of abundance from the ethanol jars. This assay is highly specific and sensitive for Dungeness crab. However, it seems to struggle when we get to about less than 10 crabs in a jar, or less than about 50 crabs in a light trap. And we believe this is due to the fact that Dungeness crab simply just does not shed a lot of DNA. In a study that compared the DNA released per copies per gram of specimen, we found that Dungeness crab is basically aligning with the lowest amount of DNA shed per organism that has been measured. And we're also seeing similar struggles in other studies. So I have a colleague who worked with Caribbean spiny lobster and could very clearly see the lobster. However, when they used their assay to apply to their eDNA water samples, they could not detect the lobster. Also, we did apply it to some Wacoa cruise seawater eDNA samples as well, and very clearly the Dungeness crab occur there. We have some caught from there. We weren't even getting detections there. So this suggests that they just shed less DNA, and just a general challenge for the decapods. However, in terms of fisheries application, this assay is most likely very well applicable, because we can still capture that high larval abundance and get those recruitment indices, and probably this assay will most likely perform at its peak when there is peak recruitment. So some future work of this assay is applying it to some outer coast light traps, because they can have counts into the 10,000s, have long-term use of this assay with light trap count, and possibly scale up to more light trap locations, and also comparison of this assay to more data points, so having more light trap collections and water samples to compare against. The impacts of the study hope to have is to increase this data generation of the larval population numbers, and also increase our understanding of Dungeness crab recruitment, which in turn will hopefully help bolster management and sustainability of the species. Here is a QR code of my references if you would like to refer to them. I would like to acknowledge NOAA PMEL, the Ocean Molecular Ecology Group, Jackson State, the Smithsonian, the Swinomish Indian Tribal Community, the Natural History Museum, Los Angeles County, and just everyone who has supported me along this project, and my funding by NOAA CCME-II. Thank you all so much for listening.
Excellent. Thank you so much, Dwan. We're going to invite Nicole and Dwan back on camera. Okay, and we'll stop the screen share. Awesome. So if you have questions, please place those in the Q&A. And we do have one that was I will say it seems specific for one of your slides, but let's show that and see what we get. So our question was, what is the benefit of using a gBlock? Is it expected to mimic any PCR inefficiency you would get from a real sample?
Great question, Elizabeth.
Yes. Thank you so much for that question. So one of the benefits of using the gBlocks is basically we're generating that limited detection, limited quantification in essentially a I want to say basically a perfect world where it's perfectly matched to these specific primers and probes, and basically ensuring that we're getting amplification at these different copy numbers, so that if we're not seeing amplification, we can be like, okay, so it's probably more likely that the Dungeness crab aren't shedding DNA there, as opposed to our assays just not detecting those in general. And also, the benefit of using those gBlocks is so we can get that limited detection and limited quantification, so that we can then further assess the assay for its sensitivity and its sensitivity. Did I
Awesome.
answer the full question?
I can bring it up again. Let's
Yes, please.
see. Yep, let's see if I can. I might need to stop your screen share
Oh, yeah.
again so I can show it. Oh, here we go. What's the benefit? Is it expected to mimic or what you would get from a real sample? So I believe you answered it.
I do as well. Really, to echo what Don said, the gBlock, if I heard correctly, can improve our understanding of sensitivity of the assay.
Yes, and also I don't believe I mentioned that we did include an internal positive control. So if any of our samples failed in any type of way, we would see that in the inhibition of that internal positive control not amplifying properly.
Awesome. Yeah, and we have some comments in the chat about, the advantage of the gBlock is the exact concentration. That's great. Okay, and so our next question, and if you did put a question in the chat, please move it over into the Q&A panel so we can show it on the screen. So our next question is, do you think it would be possible to monitor species in the wild, i.e. flowing surface waters, or do you think eDNA monitoring at this point is limited to jar samples?
I don't think it's limited necessarily to jar samples. I do believe you can test in the wild. So we did get amplification at our light traps, which are kind of and even one of our downshore sites of the eDNA that we collected. It's more so in the fact that just in general, Dungeness crabs specifically just don't release a lot of DNA. We have seen other species have better amplification and detections in the wild due to the fact that they are releasing more DNA from the animal itself.
I'll tack on to that as well, Don. So eDNA monitoring is used in a variety of ways across NOAA. Specifically, you can check out where and how on our NOAA Omics landing page. But to get more specific, eDNA monitoring can be used, for example, in Don's project regarding identifying specific species of interest. It can also be used for general biodiversity monitoring as well, and is in many cases used in larger scale environments outside of specific sample jars. You can see more on examples on that on our NOAA Omics landing page.
Thanks, Nicole and Dwan. So we do have a question in the chat, but it isn't in the Q&A, so I won't be able to show it on the screen, but I do believe everyone can still read it. So the question is, did you also test the assay on bulk larvae in the samples from the light traps?
So if I'm understanding this correctly, they're asking if I tested this on the water samples from the light traps itself.
I hope
That's what
so.
I'm reading it as.
Hopefully they comment that that's it. But yes, we did test it on the light traps itself, and we saw that once we got to like less than about 50 crabs in the light trap itself from the count Sarah gave us, we were unable to detect the Dungeness crab. However, with the higher concentration of crabs, so like that one site that had like 4,000, one that had like 1,000, one that had like 900, we were able to detect Dungeness crab in them. It was just there was a lot of variability between the replicates from the sample site. I hope that answered the question.
I hope so too. Okay. There was a comment in the chat that came in through the Q&A panel that said we've not made any plankton smoothies, but the magma in the jars are coming in from the light traps.
Awesome. Thanks, Ron, so much for answering some of these questions. I do have a question, just a quick follow up. One of my most memorable points of your discussion was the cultural significance of this work within the North Pacific or Northwest Pacific. And I was wondering if you have any ideas where we might, in the future, see or develop citizen science initiatives to help monitor these efforts. I know that you were able to work with some tribal representatives for this work, and I was wondering if there might be suggestions where this project can develop more in regards to incorporating citizen science into these monitoring efforts.
Yeah, so I believe, if I remember correctly, they've already, the Pacific Northwest Crab Research Group, who is kind of spearheading with these light traps itself, I believe they've already sort of adopted that citizen science aspect of having volunteers come in to help man these light traps and check them because they can only be powered for so long. So someone has to go out every few days to not only check them, but also count them. And I believe also for the ones in British Columbia, those are done by the Hakai Institute. I think they also allow volunteers to help work with these light traps. I think the biggest thing would just be getting people involved with knowing what these light traps are and how they can help. So if they have a dock or a pier where they typically have Dungeness crab, megalobe, if they could allow these light traps to be docked there and they like kind of take care of it to like keep people from like messing with it because people are curious,
Mm-hmm.
and also just kind of help just take some of that labor away with having to just man it all together. And if they can help with the counts, I wasn't the best when I first went out, but that second time we went out to sample, I was really good at figuring out which one was a Dungeness crab and which one wasn't. So even helping with that counting could possibly increase it. And if this assay does become applied to this process, they could essentially become water collectors. So there's someone who's collecting that water and keeping it cold for it to then be extracted later at the lab.
Mm-hmm. Another example of where eDNA can really address some of these challenges that we're facing in the field right now regarding environmental monitoring. I think that's it. I'm not seeing any more
We have
questions.
one question. I think it's just a clarification of the earlier question. So now we can show it on the screen. And they were specifically saying not on the water, but on the larvae.
Oh, we did not test specifically on the larvae itself. I think that's what Sean was mentioning about our plankton smoothies. We have not made plankton smoothies of our larvae. We did mostly just the water itself or the ethanol we removed from those samples.
Thank you, Dwan. Okay, I think now we have hit all of our questions, and I hope. Yep, it looks like that's it.
Awesome. Well,
Nicole, turn it to you.
yes. With that, Dwan, thank you so much for presenting today. It was great to understand a little bit more about your work and the work ongoing within your partners as well. With that, thank you to all who have joined today. Our NOAA Omics Seminar Series is monthly and typically takes place at 2 p.m. on the third Wednesday of the month. You can find out more about upcoming seminars on our NOAA Omics landing page, and you can see recorded seminars on our NOAA Omics and NOAA Library YouTube. With that, thank you everybody for joining today, and thank you, Don, for presenting.